Remember, there are 10 steps in glycolysis. Glycolysis is the breaking down of glucose to provide energy. That's basically what it is. So glycolysis, the first reaction, we'll go through each reaction and show you an example of it. Okay. So reaction one, the substrate glucose is phosphorylated by the enzyme hexokinase. So we add a phosphate group to glucose. The product is going to be glucose 6-phosphate. Okay. The source of the phosphate group is ATP. So now we're using ATP to donate a phosphate group. So we're actually not creating, we're using energy to get it started. Okay, this is what's called an energy debt. You know, you got to pay money to get something started, like when you invest in a business, then it gets going and you pay yourself back. Okay, that's what this is like. You've got to initially invest energy into it to get started, and then you can make more of it as you go along. So the expenditure of ATP early in the pathways works as an energy debt necessary to get the pathway started. So it's going to require ATP to get it started. And this is glucose. Okay, we add with ATP with hexokinase is going to go to glucose 6-phosphate and give you ADP plus a hydrogen. So now we got glucose 6-phosphate. Step 2, or reaction 2. The product of reaction 1 is rearranged by the structural isomer fructose 6-phosphate by the enzyme phosphoglucose isomerase. So you change glucose 6-phosphate into fructose 6-phosphate, right? And you use the enzyme phosphoglucose isomerase. The product has an exposed carbon number 1, no longer part of the ring structure. What it does, it converts an aldose to a ketose. Remember our Fischer projections. There's an aldehyde on the top or a ketone on the top. So it converts the aldose to the ketose form. And this is what it looks like. This is glucose 6-phosphate, and the phosphate groups at the number 6 carbon. And you get fructose 6-phosphate. Fructose and glucose are only difference by the position of the OH group. Remember, that's the only difference between fructose and glucose. So that's fructose 6-phosphate. And if you look at the Fischer projection, those are the Hayward projections on top, Fischer projection on the bottom. Okay, glucose 6-phosphate is an aldose. There's your aldehyde right there. Okay, fructose 6-phosphate is a ketone. It's a ketose. Okay, so you have glucose 6-phosphate converts to fructose 6-phosphate. You're basically taking aldehyde, converting it into a ketone, and you're using phosphoglucose isomerase. Remember, those are, remember the definition of isomer? Same chemical formula, different structural formula. So it's converting. Okay, that's an isomerase. It's just converting the structure. It's not changing any of the elements. Okay? So it's converting glucose 6-phosphate to fructose 6-phosphate, converting an aldose to a ketose. Okay. The third step takes the fructose 6-phosphate. Okay, it's phosphorylated again by phosphofructokinase. So you're adding another phosphate group. Okay, and you're going to end up with fructose 1,6 biphosphate. So 2 phosphate groups on your fructose sugar. And again, you're using ATP as your phosphate source. So you're using energy to kickstart this reaction in this step also. Again, it's like an energy debt that you got to use to get the pathway started. Okay. So you have glucose 6-phosphate, fructose 6-phosphate. And again, we have the 2 phosphate groups, or the 1 phosphate group here. And you use ATP, okay? And you got a phosphate group here we originally had. We had a phosphate group to the 6 or 1 position. So now we got fructose 1,6 biphosphate, because this is carbon number 1. Remember 2, 3, 4, 5, and 6. So carbon 1 and 6, now there's phosphate groups. So if we use 2 ATPs to phosphorylate that. Okay, and we use phosphofructokinase is the enzyme that catalyzes it. Remember, enzymes end with an ASE ending. And phosphofructokinase is an enzyme that adds phosphate to fructose. That's where the name comes from. And you generate ADP because ATP went to ADP. Again, you've used energy to get that going. Okay. The fourth step in glycolysis. It's a reduction reaction. It's split into two 3-carbon intermediates. So now remember, fructose, sorry, has 6 carbons. So now you're going to form 2 3-carbon intermediates by the enzyme aldolase. Okay. So you end up with glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Originally G3P and DHAP. Okay. So this is what we're going to end up with, and we're going to use this glyceraldehyde 3-phosphate in the next step of the reaction. So fructose, 1,6-biphosphate. That's what we have. We're going to use aldolase, which are enzymes, and we're going to split it into 2 3-carbon intermediates. First one is dihydroxyacetone phosphate, and the second one is glyceraldehyde 3-phosphate. The phosphate is on the number, the phosphate group is on the number 3 carbon. Remember, the carbonyl carbon gets the lowest number. Okay. We want to eventually convert our glyceraldehyde 3-phosphate back into dihydroxyacetone phosphate also. So it's step number 5. Okay. Dihydroxyacetone phosphate is then rearranged in a second glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. Okay. So glyceraldehyde 3-phosphate is the only substrate for the next reaction. So what happens is we take our dihydroxyacetone phosphate, okay, and we use the enzyme triphosphate isomerase, and that converts it to glyceraldehyde 3-phosphate. And again, it's on the number 3 carbon. Now we've got 2 molecules of glyceraldehyde 3-phosphate. Reaction number 6, glyceraldehyde 3-phosphate. We got 2 of those now. Okay, so we take our substrate, glyceraldehyde 3-phosphate is oxidized to a carboxylic acid by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Again, there's your ASE ending. This also reduces NAD plus to NADH. So there's a transfer of an inorganic phosphate group to the carboxyl group. So the first step of glycolysis is to harvest energy, bring it in. The product from glyceraldehyde 3-phosphate is going to be 1,3-biphosphoglycerate. That's going to be a product that's being formed. So a new phosphate group attached with a high energy bond. These high energy bonds are going to use to create more ATP to generate energy for the body. The ATP is the energy currency that the body uses. Now, every step from now on is going to occur twice because we've got 2 molecules of glyceraldehyde 3-phosphate. So now we're going to double everything because we generated 2 of them in the production of it. Okay. So this is your glyceraldehyde 3-phosphate, okay, and it's glyceraldehyde 3-phosphate dehydrogenase. And you can take NAD plus and you reduce it to NADH. Okay, it's an electron transfer, and so we use an electron transfer chains to generate more energy for us. And we're going to generate another phosphate group here with a high energy bond to its oxygen. This is a very high energy bond right here. So it's 1,3-biphosphoglycerate. 2 phosphate groups, one at number 1, one at number 3. Okay. In reaction 7, now we start making energy in the form of ATP. We have 1,3-biphosphate glycerate. Okay, that's what we formed in the last step. High energy phosphate group is transferred to ADP by phosphoglycerate kinase. So now we're transferring that high energy bond, that phosphate group, back to ADP. So we're taking ADP and we're forming ATP. So we're starting to form adenosine triphosphate. So it's 3-phosphoglycerate, right? Is what we're going to end up with. And ATP, you're still having 1 phosphate group on it. This is the first substrate level phosphorylation of glycolysis. Okay, so we take a 1,3-biphosphoglycerate, okay, and we're adding ADP, and we're going to convert ATP into ATP, and we're going to change the 1,3-biphosphoglycerate into 3-phosphoglycerate. Okay, the phosphate group is on the number 3 carbon. So in step 7, we're producing ATP. Okay. So reaction 8, step number 8 in glycolysis. You take the 3-phosphoglycerate is isomerized into 2-phosphoglycerate by the enzyme phosphoglycerate mutase. So isomerize means you're going to change the structural formula. You're not changing the chemical formula. So it'd be exactly the same elements, exactly the same ratio, but now we're going to change the structural formula of it. So basically all we're doing, we're moving the phosphate group from number 3 to the number 2 carbon. So we're just taking that high energy phosphate group and we're moving it from number 3 to number 2. Okay. So remember, this is 1. The carbonyl group is always number 1, 2, and 3. So this is 3-phosphoglycerate. And we're taking this phosphate and moving it to the number 2 carbon. And we do that with a phosphoglycerate mutase enzyme. And that's an isomerization. We're making an isomer. Same chemical formula, different structural formula, just arranged differently when you draw the structure. Reaction 9. Okay. The enzyme enolase catalyzes a dehydration of 2-phosphoglycerate. So we have our 2-phosphoglycerate from our previous reaction, and now we're going to use the enzyme enolase. And it's going to be a dehydration. We're going to remove water. Okay. So we end up with phosphoenol pyruvate, which is an energy rich, the highest energy phosphorylated compound in metabolism. This is one of the highest energy bonds. So we've taken 2-phosphoglycerate. Okay, we use the enzyme enolase, and we convert that to phosphoenol pyruvate. So we're taking this, making a double bond in between these 2 carbons here, okay, getting rid of this OH and H. Okay, there's our water. And we're using these enzymes, the electrons in here to form this double bond. Okay, so we're doing a dehydration right to form an enol. Remember enols and ketols? We're using to form an enol, and it's phosphoenol pyruvate. Giving us an extreme high energy bond right there. That's our high energy bond. And the final step is number 10, reaction number 10. Okay. Final subject of dehydration, it's a phosphoenol pyruvate, is what we ended up with. Serves as a donor of the phosphorus group transferred to ADP by pyruvate kinase, making ATP and releasing water. Okay. So pyruvate is going to be the final product of glycolysis. Okay. And then we're going to say with pyruvate later on too. So a coupled reaction which hydrolysis of the phosphoester bond provides energy for the formation of the phosphoanhydride bond of ATP. So we're adding water across that bond, and we're going to take ADP and go to ATP. Remember, this test happening twice for every one that we have. So phosphoenol pyruvate plus ADP, we're going to transfer this phosphate group to the high energy bond to ATP, and we're going to form ATP, and we're going to have pyruvate left over. And those are the 10 steps of glycolysis. Fructose. Fructose glycolysis. The other one was glucose. So in fructose glycolysis, it's similar. Depending on the tissue, where it is, what enzymes there are, fructose enters glycolysis in a different way. Because you already have the fructose, you don't have to form a fructose 6-phosphate. So in muscle tissues, there is an abundant hexokinase, the enzyme, that will phosphorylate the fructose to fructose 6-phosphate. So in muscle cells, we can go to fructose 6-phosphate directly from fructose. The advantage of that is muscle cells need a lot of energy. Okay, and then the fructose 6-phosphate can directly enter glycolysis and break it down to produce energy. So it saves some steps. Fructose conversion to glucose 3-phosphate. In the liver, there's abundant what's called fructokinase. Okay? This will phosphorylate fructose into fructose 1-phosphate. Okay? The fructose 1-phosphate is cleaved into dihydroxyacetone phosphate. Remember DHAP and glyceraldehyde. by your enzyme fructose 1-phosphate aldolase. The glyceraldehyde that's formed is phosphorylated into glyceraldehyde 3-phosphate, okay, which enters into the glycolysis along with dihydroxyacetone phosphate. Okay, it enters into just a different pathway. So it's skipping several steps. It's already there using those enzymes. So then there's your fructose. Okay, yeah, fructose. In the muscle, we use hexokinase. In the liver, we use fructokinase. In the muscle, you form fructose 6-phosphate, and that goes into glycolysis directly. In the liver, we form fructose. We use fructokinase to form fructose 1-phosphate. Then we use fructose 1-phosphate aldolase to form dihydroxyacetone, and enters into the glycolysis, directly into glycolysis. Then the glyceraldehyde form, okay, reacts with triose and it forms glyceraldehyde 3-phosphate, which again enters directly into glycolysis. It just enters at the different steps where that substrate is used. So it's cutting out the first couple of steps to convert fructose directly into, you can break down fructose directly without forming glucose first. And we can do that in muscle cells and in liver cells. Okay, we're limited where we can do that. Okay, certain organs only use glucose for energy, like the brain only uses glucose for energy. We can't do those conversions. So what is a substrate level phosphorylation, which we talked about? Okay. It's one way the cell can make ATP. It takes the substrate level, that's what we break the substrate down, and we phosphorylate it. And we use that to make or produce ATP, which is a high energy phosphorylate group of a substrate, and the reaction is transferred to ADP to produce ATP. Okay, so that's our substrate level phosphorylation. We end up in phosphorylating ADP into ATP, our high energy phosphate bond. Okay. What are the major products of glycolysis? Breaking down of glucose. Okay. The major product of glucose end up with 2 molecules of pyruvate. We're broken to glucose 6-phosphate, so it had 2. Started with a 6-carbon sugar, and now we got 2 molecules of pyruvate. Pyruvate is a 3-carbon compound. Okay. And we end up with, in the form of 2 ATP molecules and 2 NADH molecules that are formed. So product of glycolysis is 2 molecules of pyruvate, 2 ATP molecules, and 2 NADH molecules. Describe an overview of the reaction of glycolysis. That's a lot of steps to overview. Okay. So breaking it down, glycolysis is a pathway involving 10 reactions. Okay. And it can be entered by fructose at the different levels. In reactions 1 through 3, you need energy. The first 2 steps require ATP to go to ADP. So reactions 1 through 3, energy is invested in the beginning substrate glucose. So we're adding energy to glucose to begin the breakdown to form energy. This is done by transferring high energy phosphoryl groups from ADP to the intermediates in the pathway. So we're taking ADP and going to ADP and transferring that phosphoryl group to the intermediates along that pathway. The product after the first 3 steps is fructose 1,6-biphosphate. Right. Fructose 1,6-biphosphate is then split into 2 3-carbon molecules that begin a series of rearrangement, oxidation reduction reactions, and substrate level phosphorylation that produces 4 ATPs, remember 2 for each one, 2 NADHs, and 2 pyruvate molecules at the end. Okay, so there's an investment of 2 ATPs in the early steps. The net yield of ATP is 2. So you invested 2 ATPs in the beginning, you produce 4, so you have a net yield of 2. So we produce 2 ATPs. Now there's going to be more with the pyruvate. We just don't do that just for 2 ATPs. Moving on to the citric acid cycle, it's going to spin it and do a lot more with the pyruvate. All right. So that's the overview of glycolysis. How do the names of the first 3 enzymes of the glycolytic pathway relate to the enzymes that catalyze? They tell you the substance that catalyze and what they do. Okay. Reaction 1 of glycolysis is catalyzed by hexokinase. Hexo is 6 carbon sugar. ASC is an enzyme. So the name tells you that this enzyme catalyzes the addition of a phosphoryl group to a 6 carbon sugar. Okay. It forms a hexokinase. Reaction 2 is catalyzed by enzyme phosphoglucose isomerase. Remember, isomerase causes an isomer. Same chemical formula, different chemical structure. This name tells us that the enzyme catalyzes the isomerization, rearrangement of phosphorylated glucose. Isomerization is just rearrangement of how they are in the structural formula. Reaction 3 is catalyzed by the enzyme phosphofructokinase. Okay. Enzyme catalyzes the addition of a phosphoryl group to the phosphorylated fructose. It goes from 1 phosphate group bonded to 2 phosphate groups. Okay. 1,6 biphosphofructose. So the regulation of glycolysis, the enzyme, the step 1 was hexokinase. Okay. And it produced glucose 6-phosphate. Now, the formation of glucose 6-phosphate goes back and inhibits hexokinase, because once you produce it, you don't want to keep producing it. And you use up ATP in the process. Okay. So if you have abundance of ATP, you don't need that step to occur. So it's an allosteric inhibition. It bonds to the enzyme at a separate site. Phosphofructokinase, step 3. The activator is fructose 2,6-biphosphate. When that's present, you need that to come across. And it's going to form, the inhibitor is going to be citrate and ATP. Again, if ATP is in abundance, you don't need that fructose 2,6-biphosphate to convert it because you have enough energy already. So it shuts it down. The pyruvate kinase is step 10. And the activated by fructose 1,6-biphosphate, that's the step previous to forming pyruvate. Again, if that's present or AMP is present on any monophosphate, you need energy. So it allows it to develop. Inhibitors acetyl coenzyme A, which is the formation from what's the 3 carbon? I'm drawing a blank on pyruvate. Okay. Pyruvate turns into acetyl coenzyme A, and that inhibits more of it to be converted and the presence of ATP. Okay. So these enzymes are allosterically regulated, which means it binds in a different site on the enzyme and changes its shape, so it's not going to bind on the active site. It's not a competitive inhibitor, it's allosteric. Remember, allosteric means it binds to a different site on the enzyme. So if this was your enzyme, okay, your shape, well, let me draw a little bit differently. If this was your enzyme, like that, this is a shape that locks into place. Okay. If the ATP or citrate binds here, it's going to bind right there. And when it binds, now you've got this complex here, this complex here. Okay. So when this is bind, it's called allosteric inhibition. It changes the shape of the active site. So now the substrate can't bind to it because something's bound in a different location. That's allosteric. Okay. Fermentation. Okay. When you ferment things, produce alcohol. From glycolysis, pyruvate is your byproduct, right? Remains for further degradation, and NADH must be reoxidized again. You might take NADH back to NAD plus so you can use it again. All right. In aerobic conditions, which means in the presence of oxygen, that's aerobic. Okay. Both will occur in cellular respiration. You're going to break down pyruvate and you're going to oxidize NADH back to NAD plus. That's under aerobic condition. In anaerobic conditions means no oxygen is present. Okay, the absence of oxygen. Fermentation reactions accomplish this. You can't use cellular respiration because cellular respiration is glucose plus oxygen goes to CO2 and water. That's your net reaction. Okay? And there's no oxygen present. So you can't use cellular respiration. You have to use fermentation. Fermentations are catabolic reactions. Remember, catabolic reactions are going to break it down, occurring with no net oxidation. There's not going to be any gain in oxidation. Major fermentation pathways, there's two of them. There's lactate fermentation and alcohol fermentation. We use alcohol fermentation all the time in making beer, making wines. We ferment grapes to make wines. That's alcohol fermentation. We put in steel cylinders so we don't get oxygen in there. Okay? Lactate fermentation. When you use your muscles, if you're a marathon runner, if you're training as an athlete, if you're training doing weightlifting a lot, okay, you deplete the oxygen from your muscle cells. So then the muscles can't use oxygen anymore because you've run out of it. Okay? You can only go so much for the muscles stop. The oxygen content runs out. You can't metabolize it anymore. The body switches to anaerobic metabolism. It keeps working, but now it switches to work without oxygen. And this is your lactate fermentation. Okay? Lactate fermentation produces lactose. Lactose in the muscle causes muscle cramps. Okay? So when you're working out and all of a sudden your body starts to cramp, it's telling you, runners call it hitting the wall. You hit the wall, lactose is produced, your muscles aren't going to work anymore. They cause muscles to cramp up. That's your body's way of saying, we're running on no oxygen, your muscles getting fatigued, and now we're going to run out of lactate too. So when lactose is produced, it causes the muscles to seize up, and it wants you to stop working out so your body can recover, regenerate the oxygen in there. So you have alcohol fermentation, like we do with yeast, and we have lactate fermentation, which occurs in your muscles. Okay, and remember, those are both pathways that are under anaerobic conditions when you've run out of oxygen and you still got to break down things for energy. Okay, so lactate fermentation. Here's what happens in the muscle. Lactate fermentation is the anaerobic metabolism that occurs in exercising muscles once they run out of oxygen. Bacteria also use lactate fermentation, like production of yogurt and cheese. Okay. The reaction produces NAD plus and degrades pyruvate. So here we go. Here's our pyruvate that was caught through glycolysis. Pyruvate goes back, okay, and it produces lactate through lactate dehydrogenase. So it takes the pyruvate from glycolysis, and in the presence of no oxygen, anaerobic, it converts NADH back to NAD plus. Can't see it ever do that. Okay. And we form lactate. And lactate inhibits the muscles from contracting. So basically it tells the body you need to stop doing what you're doing because we're running out of nutrition to supply it. And that's what causes your muscle cramps. Okay. Alcohol fermentation. Okay. Yeast fermentation, sugars, fruits and grains, anaerobically, using pyruvate again from glycolysis. So we use pyruvate again. We have pyruvate decarboxylase is the enzyme, and it removes carbon dioxide from the pyruvate, producing ethanol. Ethanol is acetaldehyde, okay, ethanol. Then the alcohol dehydrogenase enzyme catalyzes reduction of ethanol to ethanol, and we make alcohol. Ethanol is our alcohol, and it releases NADH back in the process. It oxidizes NAD plus to NADH again. Okay, so when you're undergoing alcohol fermentation, a couple of things are happening. There's no oxygen, and you're producing CO2. Okay, look at champagne. When champagne is sealed in a container, no oxygen, and it goes alcohol fermentation from the grapes. We seal it there. All those bubbles in champagne, guess what? That's it. When you open up, those bubbles come up. That's trapped carbon dioxide that you've produced. That's all. Champagne is wine with carbon dioxide in it. The bubbles. That's what makes a bubble. Okay? That's what makes your soda bubble. It's carbon dioxide. It's in your drinks. Those are the bubbles. And you get it because pyruvate is breaking down to the aldehyde and carbon dioxide. Then the aldehyde is being converted to ethanol, the alcohol. So now you got alcohol and carbon dioxide in your sealed container. And how long you leave it depends on how fermentation goes along with it. Okay? They add different flavorings, different stuff, gives you the different aging process for bourbon, for scotch, for beer, for champagne. It's all just different process. It's all the same thing. It's just different flavorings, different hops, different things being added to it to give you a different taste. But all those bubbles you're getting, all the expensive champagne, it's all the same cheap stuff, except now it's got carbon dioxide in it. That's what makes all your bubbles. Okay. So pyruvate, with pyruvate decarboxylate, breaks down. Remember here you got 3 carbons, right? Now you got your 2 carbons and CO2 being released. So pyruvate decarboxylate takes pyruvate and forms ethanol. Ethanol is the aldehyde. Then ethanol goes through alcohol dehydrogenase to produce ethanol. Ethanol is the alcohol. Okay. And ethanol is the same whether it's scotch, bourbon, beer, wine, champagne, any of them always have ethanol is ethanol. CH3, CH2OH. Okay. So how is the alcohol fermentation in yeast similar to lactate production in skeletal muscles? They're both anaerobic fermentation. They both occur in the absence of oxygen. Okay. So both the alcohol and lactate fermentations are anaerobic reactions. They have to occur in the absence of oxygen. Okay. That's why when they make alcohol, they're in these big kegs, right? These big wooden barrels, and they're sealed. And you don't open them until they're ready because you can't let oxygen in. So they have to occur in the absence of oxygen. They use pyruvate, which is our end product from glycolysis. Remember, 1 glucose at the end of glycolysis gives you 2 pyruvate molecules. Okay, we reoxidize NADH. Okay, you add oxygen back into it. So you go from NADH back to NAD plus to reoxidize it, and it's produced. That was producing glycolysis. Why must pyruvate be used and NADH be reoxidized so that glycolysis can continue? Because we need to generate that back in to use it again for the reaction. Okay, the pyruvate must be used because they're allowed to build up in the cell. The last react glycolysis would stop. Okay? So you wouldn't continue glycolysis, you wouldn't continue breaking down glucose to provide energy because you're going to build up too much of the product. Remember the allosteric inhibition? The too much glucose build up, block the enzyme, and you would stop producing energy. This would cause the entire pathway to stop and no more ATP would be made. So you've got to get rid of the pyruvate. You normally use aerobic, but under anaerobic conditions, we still got to get rid of it. Okay? I mean, even if your muscles are working and you're working out hard, you're using up your glucose, using up your energy, producing pyruvate. If you don't break down that pyruvate, it's going to shut everything down. That's what goes to fermentation reactions. Okay? NADH has to be reoxidized because the cell has a limited supply of NAD+. So you got to reoxidize by taking NADH back to NAD+. If no NAD+ is available, the reaction of 6 glycolysis, reaction 6, step 6, glycolysis cannot occur because you need AD+ to transfer that electron in that glycolysis reaction. So as a result, glycolysis and all ATP production will stop. If you stop producing ATP, that's not a good thing. Remember, the muscles, if you had anatomy or physiology, remember a muscle requires 2 things to contract. It has to have calcium and it has to have ATP. Both those are required. If you stop producing ATP, your muscles will stop contracting. Your heart is a muscle. You don't want your heart to stop contracting. That's not a good thing, unless you really don't like somebody. Okay, but the contraction of heart muscles is really important. So you don't ever want to stop the production of ATP. That's why it catalyzes that pyruvate to get rid of it, so the reaction continues, whether there's oxygen or not. The pentose phosphate pathway. Pentose is a 5-carbon sugar rather than a 6-carbon sugar. All right. So the pentose phosphate pathway is an alternative to glycolysis for glucose oxidation. So in stage 1, the oxidative stage, NADPH for biosynthesis is produced. So now you're producing NADPH, phosphate with a hydrogen on it. In stage 2, 3-ribulose 5-phosphate results. Now you got a 5-carbon sugar with a phosphate group on it instead of a 6-carbon sugar. In stage 3, the ribose 5-phosphate and 2 xylose 5-phosphates are produced, along with 2 fructose 6-phosphates and glyceraldehyde 3-phosphate. So you're producing fructose 6-phosphate and glyceraldehyde 3-phosphate. Both are part of the glycolysis step pathways, and it can feed back into glycolysis at the appropriate step. Goes back into glycolysis, SIS. Okay, so you're producing 2. Remember, you're cutting it into pieces. You're putting 2 xylose 5-phosphate, and you're producing 2 fructose 6-phosphate. Okay, and a glyceraldehyde 3-phosphate and a fructose 6-phosphate. So the non-oxidative stages produce sugars with from 3 to 7 carbons on it. And the ribose sugar we need, because ribose is the sugar in DNA and RNA, which we talked about. Deoxyribonucleic acid is DNA. Ribose nucleic acid is RNA. Both use a 5-carbon sugar for the synthesis of our genetic material, for our nucleic acids. So this is a pentose pathway. Okay, the 3 glucose 6-phosphates. Okay, it takes 6 NADP pluses, goes to 6 NADPHs, and carbon dioxide. Then it goes first to 3 ribose 5-phosphate, goes to the ribose 5-phosphate, 2 xylose, 2 of the xylose 5-phosphates. And that's going to end up producing 2 fructose 6-phosphates and glyceraldehyde 3-phosphate. Both steps or both substrates in the glycolytic pathway. And then these enter back, these 2 enter back in glycolysis to produce energy. So that's how we utilize 5-carbon sugars. Now, the opposite of glycolysis is gluconeogenesis. Gluconeogenesis is the making or the synthesis of glucose. So now we're going to make glucose. Instead of breaking it down to provide energy, now we're going to make glucose so we can store it. So gluconeogenesis, this makes glucose from non-carbohydrate starting materials. So from non-sugars, this makes glucose. Okay? Like lactate, glycerol, most amino acids can do it except for lysine and leucine. Glycerol and amino acids are used only in starvation conditions. So your body has no sugar, and you have to make sure because your body's in starvation. In order to survive, you start to break down glycerol and your amino acids. The body needs amino acids. Okay? It has to have those. In times of starvation, when you've got no energy, you will break those down to produce glucose because the body needs glucose more than it needs amino acids. Continued starvation results in death. Okay? And the amino acids are not going to be used in that with the genetic material. Okay? But you're only breaking down amino acids, okay, in starvation conditions. Process occurs primarily in the liver. So in the liver is where we make glucose. So when you're dealing with patients, say with diabetes, you have to make sure and monitor glucose, liver enzymes and liver enzymes, because if you got a lot of sugar, it might not be the patient's diet bringing it in or the lack of insulin. It may be the liver is producing glucose at an alarming rate. Okay? That's why a lot of diabetics should tell your patients to eat meals frequently, not just 3 big meals a day, but 5 meals a day, smaller meals. That way it keeps the blood sugar levels up for the insulin to work and does not go into any kind of starvation routine where the liver starts to produce glucose, because sometimes the liver overproduces it and your glucose levels go through the roof. Okay, so there's lots of things that you've got to do with managing diabetic patients. There's a lot of things to consider, and this is one of them. You've got to remember when you're dealing with a diabetic patient, okay, you've got, excuse me, you got to watch the liver enzymes, because it might not necessarily be their diet. It might be the liver going crazy producing glucose and dumping blood into the stream when it gets kicked on. And having smaller meals spaced throughout the day, it doesn't kick the liver on to generate glucose. So comparison of glycolysis, which is the breakdown of glucose, and gluconeogenesis, which is the formation of glucose. Okay? So they basically opposite processes. Okay? It's not just reversing the reactions because there's steps involved in glycolysis that are irreversible. So you can't just write it backwards, we can't go through it from 10 to 1. We went from 1 to 10 for glycolysis. You can't just write the steps from 10 to 1 and go, oh, we're done. That'd be too easy, okay? Going backwards, you can't do because there's several steps that are irreversible. You cannot go backwards. Once they're formed, they can't get put back together, okay? So there's 3 non-reversible steps of glycolysis. So you got to find a new way around that. From pyruvate, you can't go from pyruvate back to phosphoenolpyruvate. That was step 10, 9 or 10. I think it was step 10, where phosphoenolpyruvate went to pyruvate. That can't go backwards. Okay. Fructose 1,6-biphosphate cannot go back to fructose 6-phosphate. And glucose 6-phosphate cannot go back to glucose, a lot of times because of the high energy bond that's been transferred. So those 3 steps you cannot reverse. Okay, these only go in this direction. Phosphoenolpyruvate goes to pyruvate, fructose 6-phosphate goes to fructose 1,6-biphosphate, and glucose goes to glucose 6-phosphate. They can't back those steps up. We don't have enzymes to do that. So now I got to make up new steps. The body has to do that. So these are your irreversible steps that you bypass. Step 1, step 3, and step 10. Okay? So you can see you can't back in those reactions. Okay? So now we're gonna have something different going backwards. We're gonna run different reactions to go backwards in that. You can use different enzymes for those to occur. Okay. The regulation of gluconeogenesis. What causes us to make glucose? So step 3 of glycolysis was catalyzed by phosphofructokinase, stimulated by high AMP, ADP, and phosphate levels. Okay. If you've got a lot of AMP, you need to make ATP, okay, because you adding monophosphate. Okay, so you need to make phosphate, so that catalyzes the reaction. It stimulates it. What inhibits it is high ATP. If you've got a lot of ATP, you don't want to keep making more. You don't need to spend that energy making more if there's a lot in the system. Okay, so that's your glycolysis. Reverse in gluconeogenesis is fructose 1,6-biphosphate is stimulated by high ATP levels because we're backing it up now. So you're going to stimulate fructose 1,6-biphosphatase because you have high ATP. So now you're going to form, you take that energy and now you're going to go back and form glucose. So you can use it later on. So at times of excess energy means high ATP levels. Gluconeogenesis is favored. Okay? So when there's high amounts of ATP floating around, gluconeogenesis is favored, glycolysis is inhibited because you've got a lot of energy floating around, high ATP levels. We're talking about the Cori cycle. In the Cori cycle, lactate, remember the lactate cycle? From skeletal muscles is transferred to the liver, converted to pyruvate, then to glucose. Okay, so here's there's your muscle cells. Muscle cells produce lactate. Okay. Lactate is produced, go back to the liver. Liver breaks it down to pyruvate, glucose 6-phosphate, and forms glucose. Okay. So in times of high ATP levels, okay, or during starvation when you're between meals, what happens in a lot of patients is the lactate from the muscle cells go back and produce glucose. Okay. Glucose is then used by the muscle, breaks down to pyruvate. Pyruvate goes back, and then it follows the sequence where pyruvate goes to lactate and goes back and forth. So if your muscle is using up glucose, you're forming pyruvate, goes to lactate, lactate goes in the liver, you produce more glucose, glucose goes back to the muscle cell. Okay. So glucose is where lactate comes to pyruvate, pyruvate goes to glucose 6-phosphate, and there's in glycolysis, and there's a backwards reaction, and goes back in gluconeogenesis to form glucose. So glucose is formed in our liver. Okay. So what are the major differences between gluconeogenesis and glycolysis? Gluconeogenesis, you form glucose. Glycolysis, you break down glucose to form ATP. Okay. So gluconeogenesis is a synthesis or formation of glucose from a non-carbohydrate source, like pyruvate. Remember, your sugars are 5-carbon, 6-carbon sugars. Pyruvate is a 3-carbon compound. So it's non-carbohydrate source. It appears to be the reverse of glycolysis, the first stage of carbohydrate degradation. Because of the intermediates in the two pathways are the same. They share glucose 6-phosphate and they share other intermediates. But reactions 1, 3, and 10 of glycolysis are irreversible. You can't go backwards. So the reverse reactions must be carried out by different enzymes. So we use 3 different enzymes to form glucose. You can't just back up glycolysis. That'd be too easy. They need to learn one thing. Okay. What do the 3 irreversible reactions of glycolysis have in common? Okay. Each of the 3 reactions, okay, the 3 irreversible reactions of glycolysis all involve, okay, high energy phosphoryl group transfers. Okay. All the irreversible reactions transfer a high energy bond. You can't untransfer a high energy bond. Okay. When you do that, you generate ATP and you generate a different molecule. You cannot back that up once it's phosphorylated. It doesn't give you the same compound anymore. Glycogen synthesis, okay, and degradation. So glycogen in the human body is polymers of glucose. It's glucose molecules linked together in a long chain with branches on them, and that's how we store glucose in the body. We store it as glycogen. Different forms for plants, different forms for animals. But if we're going to store glucose, if you have too much glucose in your system, you need to store it. We convert glucose into a glycogen molecule. It's just a linking of all these glucose molecules together. So glucose is the sole energy source. Okay, so it's a sole source of energy for red blood cells and the major source for the brain. So when making red blood cells and using on the brain, we use glucose. Okay? Glucose is supplied in our diet. We eat it. Glyconeogenesis, we form glycogen, stores our glucose for storage, and gluconeogenesis. Okay? Those are the 3 ways we can get glucose. Like I said, the brain needs glucose and the formation of red blood cells need glucose exclusively. So we got to get it either in our diet by eating it. We got to form it by gluconeogenesis, like we talked about, or we form glycogenolysis. Okay, we break down glycogen to form glucose. In other words, glycogenolysis is glucose, 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 glucose. So here's how glucose is stored as glycogen. We break these off and we release glucose back into the bloodstream. So we break down glycogen. So glycogenolysis. Okay, so by the diet, breaking down glycogen, or forming glucose. Those are 3 ways we can get glucose into the bloodstream that we need. Okay, the structure of glycogen. Glycogen, all these little red dots here, is a glucose molecule, and they're linked together, and it's formed in these branched chains. They have these long chains, and they have branches off these chains. That's glycogen. That's how we store glucose in the body. So it's a highly branched alpha 1,4. So it's an alpha linkage, which means the oxygen goes down. Remember from the glucose sugar. Okay, oxygen goes down. H goes up, OH is down. The link forms right here. So an alpha 1,4 linkage and an alpha 1,6 linkage when it bends, when it forms a branch. So alpha 1,4 forms our line. Alpha 1,6 branch forms the branches off of that. Okay. It exists as granules found in cytoplasm of the liver and in the muscle cells. That's where we store glycogen. Glycogen is our storage for glucose. Okay. Glycogenolysis. Okay. Glycogen degradation. So we break down glycogen to produce glucose back into the bloodstream. So we've stored it when we need it. When we need it, we start to break down our glycogen storage. Okay. It's controlled by glucagon and epinephrine. Remember the pancreas. Pancreas has 2 hormones that it produces. It produces other digestive enzymes, but it also produces two endocrine hormones. Sorry. Okay. The pancreas produces endocrine hormones in the form of insulin and glucagon. Insulin lowers blood sugar by taking glucose out of the bloodstream, forming glycogen. Glucagon increases blood sugar by breaking down glycogen. Okay, and epinephrine. Okay, that's formed by your adrenal gland. Okay, and that forms your fight or flight response. When your body's preparing for a flight or flight response, epinephrine is released. Your body's all keyed up. We're releasing glucose, so the muscles have plenty of glucose to respond to fight or flight. Okay, so those two hormones control the amount of glucose in your bloodstream. Well, basically three, but insulin lowers it. The two that raise it by breaking down glycogen is glucagon and epinephrine. Epinephrine is adrenaline. Same thing. Okay. So in step one, the first step, glycogen phosphorylase. Okay, this is your enzyme, glycogen phosphorylate, catalyzes removal of an N-glucose as glucose 1-phosphate. So it goes to the glycogen chain, goes there and breaks off one of the N-glucose molecules. Okay. Debranching enzyme catalyzes removal of the last glucose at an alpha-1,6 branch as glucose. So the debranching enzyme removes the glucose, then as glucose. Phosphoglucomutase converts glucose 1-phosphate to glucose 6-phosphate. Okay, those are your three steps. So here's what glycogen looks like. In the end, they're all glucose molecules. And it goes off the branched one and it takes away the last glucose. Okay. So that's glycogen phosphorylase. Goes to glycogen plus N-glucose 1-phosphate. How many times it breaks it up? So this produces glucose 1-phosphate. As it breaks it down, as it pulls off each glucose molecule. And you get glucose 1-phosphate. Okay. And that's glycogen phosphorylase that breaks down glycogen to produce glucose 1-phosphate. Okay. Then glucose 1-phosphate, okay? Or if you break it off from with glycogen, if you're going to use a glycoside, which is a debranching enzyme, it forms glucose directly. So depending which enzyme you're using, glycosidase is a debranching enzyme, breaks down the branch. If it breaks it off at the branch, then it goes directly to glucose. If it doesn't, it goes to glucose 1-phosphate. Okay. And then glucose 1-phosphate goes to glucose 6-phosphate by phosphoglucomutase. So it takes your phosphate group and moves it from the number 1 carbon to the number 6 carbon. So you have you break down from the N glucose on the chain, the branch, you break that off, you end up with glucose 1-phosphate. Then with phosphoglucomutase, you convert that to glucose 6-phosphate. If you break it down from the branch by separating the branch from the main chain, okay, then you get directly form of glucose. Okay, so explain the role of glycogen phosphorylase in glycogenolysis. Okay. So the enzyme glycogen phosphorylase, which we talked about, catalyzes the phosphorylation of a glucose unit at the end of the glycogen molecule. So the glycogen molecule at the end, it puts a phosphate group on the glucose unit. Okay. It involves the displacement of glucose by a phosphate group. So it puts a phosphate group where it was bound to the glycogen molecule. And it produces glucose 1-phosphate. Okay. And then the glycogen molecule is 1 glucose unit shorter. So it takes it off from the branched end, uses glycogen phosphorylase. Okay. It takes the glycogen molecule, snaps off that 1 glucose, puts a phosphate group on it to break the bond. Okay. And it produces glucose 1-phosphate. And then it's converted to glucose 6-phosphate. Okay. So how does the action of glycogen phosphorylase and phosphoglucomutase result in an energy saving for the cell? if the product glucose 6-phosphate is used directly in glycolysis. Okay. And what happens is glycogen phosphorylase, like we talked about, catalyzes the reaction in which glucose unit is simultaneously removed from the glycogen polymer, and a phosphoester bond is formed between the hydroxyl group of C1 glucose and of the inorganic phosphate group. Okay. Phosphotrin phosphorylase produces glucose 1-phosphate. Just like we said in the last question, glucose phosphorylase attacks or breaks the bond from the glycogen molecule where the branched end of the glucose is severed, and it says by adding a phosphate group there, and it phosphorylates and forms glucose 1-phosphate. This is what's being formed, and you're branching off with glucose glycogen phosphorase. Then you have glycogen left over. That's 1 glucose shorter. Okay, phosphoglutamate mutase catalyzes the reaction for isomerization. Remember, isomerization means you're changing it the structural formula, not the chemical formula. Same chemical formula, different structural formula. Okay, then you go to glucose 1-phosphate. This converted to glucose 6-phosphate. Glucose 6-phosphate is able to go into glycolysis and break down and form energy. It's very efficient because it does not require the expenditure of energy. You don't have to use those 2 ATP molecules in the beginning to get it started. You've already done it when you broke the bond from glycogen. Okay, so you don't have to do the first 2 steps that use up ATP. Okay, so it's more efficient that way. Okay, it does not require the expenditure of energy in the form of a hydrolysis, a phosphoanhydride bond of ATP in order to phosphorylate the glucose, because it's already phosphorylated. You did that when you broke it apart from the glycogen molecule. Okay. Glycogenesis, now formation of glycogen. That's how you break down glycogen for energy. Now, how do we form glycogen? Glycogen is formed, glycogenesis happens when there's too much glucose in the bloodstream. We have more glucose than we need. So insulin stimulates the synthesis of glycagon. Okay? Glycogen is stimulated by insulin because it removes sugar from the bloodstream by lowering the blood sugar. Glucagon increases the blood sugar, or breaking down glycogen. Insulin stimulates or lowers our blood sugar by causing the synthesis or formation of glycogen. Okay? So glucose plus ATP, okay, goes to glucose 6-phosphate plus ADP plus hydrogen. The enzyme involved in that is glucokinase. Then glucose 6-phosphate goes to glucose 1-phosphate, okay, by the enzyme phosphoglucomutase. So glucose must be activated to add to the growing glycogen chain. There has to be something, when it bonds, it can split that bond and break apart and add water between there so it can link to the growing chain of glucose molecules in glycogen. So glucose 1-phosphate plus uranine triphosphate goes to uranine diphosphate, glucose, which is bonded to it, plus your inorganic phosphate group. So UDP, uranine diphosphate glucose, adds to the growing glycogen chain. And that's how it does it right here. This is reaction of glycogenesis, formation of glycogen. So here's our glucose. That's what we start with in the bloodstream. Insulin is released, it catalyzes this reaction to start, and it reacts with ATP, and now you got a phosphorylate group on your glucose. So it's got glucose 6-phosphate. Okay, and we take our glucose 6-phosphate, and we use phosphoglucose mutase to move this phosphate group to the number 1 position. So now we form glucose 1-phosphate. Glucose 1-phosphate, okay, we use that phosphate bond to link between here to form this linkage, okay, with the UDP to form urine triphosphate. So it links that together with a high energy bond. Okay, so we went from glucose 1-phosphate plus UTP. We use the enzyme phosphorylase, okay, to produce UDP glucose, okay, urine diphosphate, uridine ditriphosphate, and pyrophosphate. Those are your inorganic phosphate group. Right? So now we got a high energy bond here, and we can use that to add that to glycogen. Now it's ready to add to glycogen. We can add another glucose molecule to our growing glycogen chain. So there's the different reactions. We have glucose. This is glucose right here. This is our phosphate linkage. Okay. And this is a urine diphosphate. Okay, that's our nucleic acid urine. Okay. I meant to include. This is your urine diphosphate. There's your two phosphate groups, and that's your uridine molecule. Okay. Once that's it, now it's activated and primed to add to our glycogen chain. Okay. So the action of the branching enzymes. Okay, we're adding to our chain, which are 1,4 linkage. Okay, then we have a 1,6 linkage, which forms a branch. So we keep building our chain by adding glucose molecules, and then it branches. Keep building our chain, and it branches. Keep building our chain, and it branches. And then the branches can branch too. Okay, so you get this multi-layered branching occurring in our glycogen. So glycogen is a branched storage molecule for glucose. And the 1,4 linkage, 1,4 glycosidic linkage, the alpha 1,4 is carbon number 1 bonds to carbon number 4 of the next one. Okay, which is because here's your number 4 position. And this is, didn't draw that very well. Okay. This is a number 1 carbon, remember? This is a number 4 carbon. 1, 2, 3, 4. So it's a 1,4 alpha linkage. Remember, alpha is down, beta was up. So it's an alpha 1,4 linkage. Okay. When you have a branch on it, you have an alpha 1,6 linkage. Here's your number 1 carbon, and this is a number 6 carbon. So if it's an alpha 1,6 linkage, it forms a branch, comes off at an angle. If it's an alpha 1,4, it just forms a linear. Okay. So every branch that you have is an alpha 1,6 linkage. Every addition of glucose that's in a linear or straight line, this is an alpha 1,4 linkage. And that's what determines the branching of glycogen. Okay. So describe the way in which glucokinase traps glucose inside liver cells. Okay. Because remember, liver is where the glucose actually is converted into. Okay. Glucokinase traps glucose within the liver cells by phosphorylating. It adds a phosphate group to it. Remember, that's an irreversible reaction because the product glucose 6-phosphate is charged. It cannot be exported from the cell. So once you put that phosphorylation group on it, it doesn't get kicked out of the liver cells. Because you remember, the membrane of a cell is nonpolar. Once we put glucose 6-phosphate, we had a phosphate group. We've added that to it. Now we put a charge on it. We put a charge on it, it can't cross the membrane. So it stays within the cell. So it traps it within the liver cell. Remember, liver cells are producing glucose from the glycogen, breaking it down. Describe the reaction catalyzed by the branching enzyme. We just did that. The branching enzyme removes a section of alpha 1,4. Okay, well, if you link glucose units from your glycogen polymer, which is your long chain, and reattaches in the 1,6 linkage. Okay, so 1,4 produces your linear one. It removes a 1,4 linked one, and in its place produces a 1,6 linkage, an alpha 1,6 linkage, which causes a bend or a branch in the chain. Glycogenesis and glyconeolysis. Okay, the formation of glycogen versus the dissolving of glycogen. So when you have patients, you can have patients with hyperglycemia. Hyperglycemia is high blood sugars. Remember, high blood sugars can be caused by diabetes, okay, or it can be caused by the liver producing too much glycogen, too much glucose. Okay, insulin stimulates glucose uptake. Okay. So insulin causes the glucose to be taken out of the bloodstream and put into into glycogen. So it elevates glucokinase, it activates glycogen synthase, and inhibits glycogen phosphorylase. So it elevates your glucokinase, okay, to phosphorylate your glucose. It activates your glycogen synthase, which forms your glycogen linkage, either your alpha 1,4 or alpha 1,6. And it stops glycogen phosphorylase from phosphorylating glucose and putting it into the glycolysis pathway. Okay, so that would keep it in the blood. So basic effects to get it out of the bloodstream and store it. Low blood sugar is hypoglycemia. Okay, that'd be the exact opposite. Okay, low blood sugar, you have glucagon. Glucagon is going to raise your blood sugar. Glucagon stimulates glycogen phosphorylase, which is the exact opposite. Okay, exact opposite of insulin. Insulin inhibits it. Glucagon stimulates it because you want to put sugar back in the bloodstream. Okay, and it inhibits glycogen synthase, whereas this one activates glycogen synthase. So it's doing the exact opposite of insulin, which is what we expect. Insulin is going to lower your blood sugar. Glucagon is going to raise your blood sugar. So depending on your condition, the pancreas is going to excrete either insulin or glucagon, and they work in opposition to each other to maintain a relatively constant blood sugar level. So glycogen synthesis and degradation do not compete with one another. You're not making it and breaking it apart at the same time. The different enzymes being released that either you're doing one or the other. The body's not doing both at the same time, which would defeat the purpose. So it's being controlled by two different hormones. Okay. So opposing effects of insulin and glucagon on glycogen metabolism. So insulin, okay, it inhibits the reaction of glucose, okay, from blood to the liver, and it stimulated your glucose uptake. Okay? So insulin lowers your blood sugar. Glucagon raises your blood sugar. Okay, and it's acting on the different enzymes for the reactions to occur. Okay, it goes from the liver to the blood to raise blood sugar. This goes from blood to the liver to decrease the blood sugar in the blood. Okay, so you're raising the sugar in the liver and you're using it to form glycogen. Here you're taking that out of the liver and you're forming glucose into the bloodstream because your body needs it. And it's always the opposite. So here you've got stimulation and you got inhibition. Okay, because you're going to inhibition, stimulation. You're doing the exact opposite. So insulin is taking blood, the sugar in blood, and going to your liver to store it as glycogen. Glucagon's taking blood sugar from your liver and putting the sugar levels high back into your bloodstream. Explain how glucagon affects the synthesis and degradation of glycogen. Remember, insulin and glucagon work in opposition to each other. Glucagon is secreted by the pancreas and stimulates the raising of blood sugar. So it causes the liver to break down glycogen to put glucose back into the bloodstream. Okay. So glucagon indirectly stimulates glycogen phosphorylase, which is the first enzyme of glycogenolysis. Okay, the breaking down of glycogen to put glucose back in the bloodstream. This speeds up glycogen degradation. Glucagon also inhibits glycogen synthase to form glycogen, which is the first step in glycogenesis. So it stops the synthesis or formation of glycogen, and it stimulates the breaking down of glycogen into the glucose molecules back in the bloodstream. Okay. How does insulin affect the storage and degradation of glycogen? Exact opposite. Insulin is released in response to high blood sugar levels. Insulin reduces the amount of sugar in the bloodstream. Okay. So insulin increases the storage or the formation of glycogen and decreases the breakdown of glycogenolysis, the breaking down of glycogen. So insulin activates glycogen synthase, which is the first agent of glycogenesis. Okay, it increases the production of glycogen. It takes glucose out of the blood, puts it in the liver, and then puts it in the form of glycogen. This stimulates glycogen synthesis and storage. It inhibits glycogen phosphorylase, which is the enzyme glycogenolysis, which breaks down glycogen to put more glucose in the bloodstream. So it inhibits glycogen degradation. So they work exactly opposite of one another. High blood sugar, insulin's released to lower the blood sugar in the bloodstream. Blood sugar is too low, glucagon is released to break down the glycogen to put more sugar back into the bloodstream. both released by the pancreas.